Hey, this is Dr. Guerrero. I just wanted to walk you through our pH and macromolecule labs, the results, in case maybe you didn't get to it or perhaps you didn't understand how the answers were to be interpreted. And this is also, of course, to review you for your upcoming lab exam. So the labs that we did in class, we did 4.2, which was the pH lab. And then we did lab 5, but there were 3 aspects to lab 5, A, B, and C, where we tested for various macromolecules or biomolecules in some common foods. So I'm going to start with 4.2, which was the pH lab. We took water, milk, orange juice, detergent, non-phosphate detergent, and vinegar. And we took their initial pH with the pH paper and you recorded it. Then you added 1 milliliter of vinegar, and then you added 2 milliliters of vinegar and 3 milliliters. Yeah, another 2 milliliters, so that it's that 1, 2 is 3 plus 2 is 5. So you kept adding vinegar. And what we were looking for is if there was a change in pH. And I do want to remind you that a change in pH, or pH, is just a measure of hydrogen ions, the amount or the concentration of hydrogen ions. And when we interpret pH, we interpret it as acidic or basic. So if it's acidic, it falls on the pH scale below 7, so smaller numbers. And if it's basic, it falls on the pH scale above 7, so larger numbers. And we talk about how strong or weak those acids or bases are based on the amount of hydrogen ions that they have. Basically, what you should have seen is water should have started off with a kind of a neutral pH. And then as you added vinegar, it became more acidic. Milk should have started off as kind of an alkaline or a basic pH slightly. And then as you added vinegar, it became more acidic. Okay, orange juice pH should have started off acidic, and then if you added vinegar, additionally, detergent, vinegar would have been the control, so it didn't matter how much vinegar you added, it would still have the same pH each time. Okay, so remember that pH is the measure of hydrogen ions, and it is interpreted as either acidic or basic. Okay. So then we did 5A, and 5A had 2 parts. 5A1, we tested for simple sugars. Simple sugars fall into the macromolecule or biomolecule of carbohydrates, and the indicator that we used was the Benedict's reagent. We took the following foods and we put them in test tubes, and we labeled those test tubes, and we added Benedict's reagent to each of them to test and see if simple sugars were present. So I have kind of an outline of what the results could have looked like and how we would interpret those. Okay. So first of all, knowing that Benedict's reagent is the indicator for simple sugars. And so it's only going to test positive for like monosaccharides like glucose, fructose, maybe some disaccharides like lactose. All right. So we always test in water because water will serve as our control. We know water has no carbohydrates. So whatever color the water turns, we know that that is a negative result. Okay. Then in class, I talked about perhaps hypothesizing which of these did have sugars. Okay. As you go through and you test and you add your Benedict's reagent, you may have a gamut of colors here that have shown up. And the color change depicts about how much simple sugars are present. Okay, so you can see here in the conclusions that I have plus signs and then maybe 2 plus signs, meaning that this has more simple sugars than one with just a single plus sign. And like fructose and glucose, excuse me, should for sure be made up of simple sugars. Making sure, again, you know the Benedict's reagent, and you know what the color change is, and you know what we're testing for, okay? The second part of 5 was we were testing for complex starches. Well, starches are carbohydrates, okay? And the indicator that we used for this one was Lugol's iodine. So again, we took different foods and we put them in our test tubes, labeled our test tubes, and then added Lugol's reagent, Lugol's iodine so that we could test to see if complex sugars were present. Complex sugars or starches are present. The color change here is indicated. So here is a negative test, and here would be a positive test, and we would expect to see different degrees of darkness depending on the starch is present, the amount of starch is present. We always test on water because it's our control. All right, and it tells us what happens when there are no starches present. Another one that we use as a control or an indicator would be testing starch itself. We would expect starch to come back positive, right, for starch. And so here are some conclusions, but making sure you know what you were testing for. So complex starches, that you were using Lugol's iodine, and what the color change was. Part 2 was the lipids test. There were 2 lipids tests that we ran. The first one was a brown paper bag test. So we took just these foods here, and we took a drop of them and put them on a brown paper sack and then let them dry. If they dried and there was no stain, then they were negative for lipids. But if they dried and there was a stain or it became transparent, we knew they were positive for lipids. So based on this, we knew that water would be our control, and it dried, so it was negative. And we would expect for sure vegetable oil to be a lipid because oils are a lipid. So it stained or it made it transparent, and so we knew that that was positive. Okay, again, knowing what we were testing for, lipids, what we tested on, or what indicators, so the brown paper, and what a positive and negative reaction look like. The second lipids test was B2. And in B2, we took our test tubes now instead of brown paper, and we used the indicator Sudan 4. Sudan 4, we put our test tubes, set them up, labeled them, and then we added the Sudan 4 and looked for a reaction. Here is kind of what a negative reaction would look like, and here is a positive reaction. Again, you may have some slight variations of this, but ultimately this is what we're looking for. Also, when lipids are present, there tends to be a separation because you've put distilled water in here as well. So you may see some bubbles forming or like oil and water separation. So be aware of that. We obviously tested water first. It was our control. We knew that there were no lipids present. So when we added Sudan 4 to water, we got this. So we know that that's what a negative test looks like. Also, just like we did with the brown paper, we knew vegetable oil would for sure have lipids. So we set that up, added our Sudan 4, and we see what it looks like when it tests positive for lipids. Okay, then you test your other solutions that are listed and decide whether they are negative or positive for lipids. Again, knowing what you're testing for, lipids, what you're testing it with, Sudan 4 is my indicator, and what the positive and negative results look like. The last test that we ran was the test for proteins. The indicator that we used here was Biuret reagent. It may also be called Biuret or Biuret reagent, however you want to say it. This is a indicator that tests for the presence of proteins. So we took our test tubes, we labeled them, and then we tested these different foods. Of course, we started with water first. Knowing that water was our control, when we added Biuret reagent to it, we knew that this would be the color indicator for something that was negative for proteins because water doesn't have proteins. So we recorded that color reaction. Then as we tested these other ones, we looked for a color change. One of the foods that we talked about being for sure positive for proteins was egg whites or egg albumin. And so when you set that up and you added the Biuret reagent to it, it turned this dark purple color. So we knew for a fact that was what a positive protein test looked like, and this is what a negative protein test looks like. Again, controls are really important when you set up your experiments so you know what positive and negative look like. Outside of that, remember protein test, Biuret reagent, and what a positive and negative result look like. I appreciate you taking time to review for this exam. Best of luck. Make sure you reach out to me with any questions.